Hello. I've got a three-parter for you today. We're going to talk about PCR, then we're going to talk about the way that you can quantify the amount of PCR products. It's called quantitative PCR or qPCR. And then we're going to talk about reverse transcription qPCR, just adding one more step on. And that's going to be where we want to end up, and that's what I'm going to change the test question to reflect. So let's talk about PCR, quantitative PCR, and quantitative reverse transcriptase PCR. All right. So PCR solves the problem of how can you identify a specific sequence from a genome and copy it. Now, copying is the first step to a lot of different things. When you have a lot of copies, you can read out what the sequence is. You can also save a copy, which is called cloning. You can put it away inside of a bacterium and just keep it essentially forever. You can also manipulate that copy. So you can take that gene and move it from one animal to another, or express it in a different kind of cell, or do whatever you like with it at that stage. PCR is a useful technique. This is the way they do paternity testing, and it's also the way they would test for a lot of diseases. So, for example, a COVID test, which is actually looking for the genome of SARS coronavirus 2, is going to be done by quantitative reverse transcriptase PCR. So that's what we're going to build up to. And it's not only useful for this. You'll find it very commonly in hospitals. This is a good way to detect different kinds of metabolic disease. And if we're talking about personalized medicine, where we actually tailor the type of treatment to a person's genetic makeup and the way they are using their genes, then a tool like quantitative reverse transcriptase PCR becomes absolutely essential. So this is really widespread, used for a lot of different diseases, and this is a thing that I think you ought to know about. So let's go there. All right. And I'm going to use two sets of slides from the company Biol, which I think owns some of the patents on the PCR enzyme we're going to use. And one last little slide set of, I think, 4 slides, which is one that I've put together. Okay, so the problems that you're going to run into with PCR. One, there's a lot of DNA out there. And a genome is gigantic, and usually you're just interested in a little piece. Genomes can be billions of nucleotides long, but a gene is usually only going to be a couple thousand nucleotides at the most. Yeah, and a lot of those are going to be smaller. So we have to specifically pull out the right part of the genome in order to copy it or read it or do whatever we're going to do. The other problem there is that all DNA is only made of the nucleotides A, C, G, and T in some order. And there's only so many ways you can arrange these things. So it gets difficult to tell one little patch of A, C, G, and T's from another one, conceptually. But PCR does solve this. Basically, if you get a long enough string of A's, C's, G's, and T's, you can then check it against all the other known sequences in the world, and you can make sure that yours is actually going to be specific and is going to bind to one particular target above all others. And if you can do that, then you are, you've solved half the problem facing people who want to work with genes. The other problem is that there are usually only 2 chromosomes inside of a cell. The chromosome from the mother's side, the chromosome from the father's side, and with chromosomes like an X and a Y, there can potentially only be one of that chromosome inside of every cell. So if you want to do anything with these genes, you need not just millions of copies, but more like trillions of copies minimum in order to be able to do pretty much anything with a sequence. And so what we have to do is take a small amount of DNA and copy it, which is called amplification. And PCR does both of these things really well. It specifically amplifies sequences. All right, so yeah, yay PCR. Okay, PCR is invented by a guy called Kerry Mullis. I got to hear him talk a couple of years back. I don't know. On one hand, he comes off as crazy as a loon, but on the other hand, you can tell he's fairly smart. He would tell you the story if he was here about him driving along the Pacific Coast Highway and it just coming to him, almost like in a vision as he's driving his convertible. And this is very fortunate because the place where he was working, if he was on site when he had this idea, then the idea would belong to the place where he was working. And but since he had this idea in between jobs while driving a car, it was just fine, and the idea was his to do with as he pleased. So yeah, that's neat. All right. Yeah, lots of uses. Forensics, you can identify. If you have a hair, there will usually be a little clump of cells at the bottom of that hair if that hair has fallen out or been pulled out. And these are cells from the follicle of the hair. And if you got a couple of cells there, even one cell potentially, you can copy the DNA in that cell and you can match it up to DNA from, potential victims, yeah. It's a way to identify basically any sort of fluid or substance that has a little bit of human genetic material in it. And so, yeah, this is used in rape kits. This is used all over the place. Paternity testing, definitely. Because a child will have genetic markers from their mother and father. There are particular regions of the DNA that usually don't code for genes, but are these repetitive sequences. And people will have different numbers of these sequences and slightly different versions of these sequences. And so those are what they actually use for paternity testing. If you're interested in understanding more about the forensic uses for PCR, the class for you is going to be Biology 473, which is DNA forensics. And yeah, I think you'll really like that. It's something that you can take in the next couple of years. Okay, it's also useful in archaeology, even in paleontology. They can amplify DNA from long dead bodies, and you can kind of get a sense of who this person might be related to. Certain genes tend to occur a little bit more frequently in certain historical populations. And so, yeah. Some people would like to reconstruct dinosaurs from blood, but we think the longest the DNA can survive under optimal conditions is probably something like 6 million years based on the rate of decay. And so the oldest human DNA that we have is only a couple of 100,000 years old. The oldest animal DNA, I think, is only a little bit older than that, a few 100,000 years old. And I think the oldest DNA currently on record is from a plant. And I think that's somewhere around 1 million years old. So they're figuring with optimal preservation, maybe you could stretch that to 5 or 6 million years, but not back as far as dinosaurs. The proper dinosaurs die out 65 million years ago. Although birds are still around, and birds come from within the dinosaur tree. And we have a sequence of chickens. So, yeah, if you have chickens running around your yards, you have little baby, stupid-looking dinosaurs running around your yard. Fantastic. And disease. Yes, we use PCR for this and for figuring out how to treat it and for all kinds of other wonderful things. Wildlife research, you can, if you've got somebody who has illegally poached a rhino horn or a leopard pelt, you can actually sample DNA. You can get DNA back from that or from museum specimens that are 100 years old, and you can trace it back and you can figure out which population of animals it came from and probably which park it was poached from. Really interesting stuff. And then, of course, yeah, we do all kinds of work with DNA in the laboratory. That's a lot of what biology ends up being. Since this is the study of life, and life doesn't really exist outside of cells. Basically, if you want to play around with a living cell or understand a living cell, you're going to have to get good at working with DNA. All right, so how does DNA work? Like so. So you're going to take double-stranded DNA, which is how DNA comes in this world. A single chromosome is going to have double-stranded DNA on it. That's just normal. And you're going to make copies of it. So you're going to have these little tiny sequences, the red and blue thing, and each of those is just a piece of DNA called a primer. And so the first step in the process is you're going to attach the primers to the DNA. Or rather, the first step is you're going to separate the two strands of DNA into single-stranded DNA, which we've just done. Then you're going to add the primers in, let those settle down onto the single strands of DNA, And then the primer serves as a starting place. It allows the enzyme called polymerase, which is shown as a yellow oval here, to have a start. And the polymerase can't start on its own, but if a polymerase sees a start and it sees something to copy, it will just run through and copy everything until it gets to the end and basically falls off the end of the DNA. And so PCR is just the process of repeating this over and over again. So you can go from 1 copy to 2 to 4, 6, 8, 2 to 4 to 8 to 16. There we go. Doubling, you know? Like they do in math class. So first step, denature. Second step, anneal primers, which means stick them down. Third step, replicate DNA, just like that. Let's take that bit by bit and let's talk about it. So what do you need to actually do PCR? Well, these are the components, and that's going to be one of the things that I'll be looking for in a test answer. So water is just the way where the enzyme is going to work. You need a water sort of background for all this to happen in. The buffer is going to adjust the pH and the ion concentration to a point where you can actually do PCR, because you're going to be involving a enzyme called a DNA polymerase, and notice the ASE ending on the end, which means it's an enzyme. And so the rest of the name tells you what kind of an enzyme it is, and so this is a polymer-making enzyme. So a polymer is just linking up lots of little tiny things to make a big long chain. In this case, we're linking up little tiny nucleotides to make a big long chain of DNA. And so they call it a DNA polymerase, DNA polymerase. That's the way it works. Now, our DNA polymerase is going to be a thing called Taq polymerase. That's TAQ. And Taq polymerase requires magnesium ions. And so there's a little spot in the core of each Taq molecule that has a magnesium. And as the polymerase is working, that magnesium is kind of loosely held, and it'll pop out every once in a while. When it pops out, the enzyme grinds to a halt immediately until another magnesium comes along and slots in, and then the enzyme's going to pick back up. So it's kind of like those ion channels that we were talking about. Do you remember the calcium-gated ion channel that just sort of fell apart and snapped shut every time calcium was missing? And then once you put another calcium in, then boom, it would open up again. Yeah, kind of like that. The polymerase is going to need a couple of things in order to work. So first of all, it's going to need a template. Template is the DNA you plan to copy, and that makes sense, I think. Second thing you need is primers. So primers are little pieces of DNA that you've made specifically to stick to the DNA that you intend to copy. So this means that you have to know already a little bit of the DNA sequence in order to make a copy of that DNA or work with it. So this was a technique that was hard to do at the beginning when we didn't really know anything, but now that we know the DNA sequence of all kinds of creatures, it's actually fairly easy. You just look up what the sequence is before the one you want to amplify and after, and then you can copy whatever is in between. And as you're making the copy, you're going to have to make new DNA, and so you're going to need DNA nucleotides to assemble into that new strand of DNA. So the primer is going to serve as a starting point. It's a little piece of DNA that will serve as the starting point. It'll tell the polymerase where to begin, and the polymerase will extend the primer. It'll add on to the back of the primer by putting in nucleotides until it runs out of room or runs out of time, or just, enough of its magnesiums fall out that it just gives up and falls off. Okay, so let's go through all these things. Water, yeah, that's what everything's swimming around in. The buffer is going to stabilize the DNA, stabilize the nucleotides, and stabilize the polymerase that you are going to be using. So this is important. There are certain conditions under which you can copy DNA. There are other conditions under which it's just not going to happen. And so when you buy in your DNA polymerase, it's going to come with a little tube of buffer, and you just add the right amount of buffer that it says, and that makes everything work. Okay, the DNA template is what you're going to copy, just like we said. The primers are going to stick to the DNA template, and you're going to make a primer that starts before the thing you want, and a primer that starts after the thing you want. And you're going to use these primers to copy the thing you want, which is going to be in the middle. So yeah, we call these the forward and reverse primers, the one that's in front of and behind the primers. And when we design the primer, we're going to be able to calculate what is the temperature at which this primer should bind onto the template. A primer that is short will have you'll have to have colder temperatures for it to bind. The longer your primer gets, the more this is going to want to bind, and so it's going to bind, and you're actually going to be able to bind it down at a higher temperature. So if you raise the temperature up, if you cool down the temperature down to freezing, you can get pretty much any piece of DNA to stick to any other piece of DNA. And we don't want that. We want our PCR primer to stick to exactly the right part of the DNA, not just any part. So it says here 15 to 30 nucleotides is a good length. Typically, what you're going to see is 18 to 21 nucleotides. But for different purposes, yeah, you can expand it. And you can make longer primers than 30 nucleotides. That's fine. It's just expensive, and you don't really get much benefit after you go much beyond about 21, or occasionally I'll go up to maybe 24 nucleotides. And then, yeah, you're going to put in a known amount of primer. Now, you want the limiting reagent. So this is a concept from chemistry. Limiting reagent is whatever you have the least of, whatever is going to slow down the reaction. You don't want that to be primers. You don't want that to be buffer. You don't want that to be magnesium. You don't want that to be nucleotides. And you don't want that to be the polymerase. You want your limiting reagent to be the DNA template, because you don't really know necessarily how much DNA template you're putting in. And it may only be a few copies. And however many copies you get in there, you want to make sure that you are able to copy every single one of those as efficiently as possible. So the template is going to be the limiting reagent, the unknown reagent in a sense. And yeah, you're going to put in far more primers than you will ever need. And that's going to pay off down the road. Okay, nucleotides, you need your A, G, C, and T. That's just, yeah, basic biology. So here's your primer up here, and they're showing the different nucleotides in different colors. And so there's the end of the primer, and the polymerase will add on to the primer, and it'll make a copy, which is a reverse complement of the other strand. So when you have two DNA strands together, they're going to run in opposite directions. So this one's going to run, this is its front, this is its back, and the opposite strand that's bound to it is going to start here, its front is here, and its back is here. So it's like they run opposite ways. If you put your arms together like a genie, you'll notice that they run opposite ways, and that's a good thing, you know, to think of mentally as you're picturing DNA. And the two sides of DNA are not identical to each other, but they are reverse complements. So if you have AA, which are these two green ones up here in the top, you're going to have TT in the bottom, down here. And if you have a C in the top, which is the blue one, you're going to have a G in the bottom, which is the black one. Just like that. That is a reverse complement. And so, yeah, you want to put in far more nucleotides than you are ever going to use, so that nucleotides are not limiting. So magnesium, like we said, is essential for the polymerase. And the polymerase itself is an enzyme called the Taq polymerase. It stands for Thermus aquaticus, which is a kind of bacteria. And thermus is spelled with a U, thermus. There you go. And so we abbreviate that as TAQ. And so we just say that as Taq. And if you ask anybody in a molecular biology lab, oh, do we have any Taq? They'll tell you, yeah, sure. They'll tell you right where it is. This is something you need all the time. You'll use it pretty much every week if you're in some kind of lab that's working with DNA in roughly any way. And the Taq is going to be heat stable. That is its greatest property. So Taq originally comes from a hot spring, I believe at Yellowstone Park. And so you're going to be able to heat Taq up to the point where the DNA strands are going to separate. And so you're going to separate the DNA strands with heat. But since the Taq is this really strong enzyme, it's actually fine if you heat it up to 95 degrees Celsius, so just before boiling. Taq is cool with that. And that's what makes Taq work. That's what makes PCR work. And then Taq is going to have an optimum temperature. It's going to run best at about 72 degrees Celsius, which is really hot. It's something like neighborhood of 150 or 160 degrees Fahrenheit. Yeah, you can do the calculation yourself if you want to know exactly what. But nobody in science really uses Fahrenheit except to translate for non-scientists what they're talking about. So I don't know what it is, but yeah. Some versions of TAC will run best at about 68 degrees, others run best at 72 degrees. Okay, so those are the parts that make a reaction. And then you're going to take all these things and you're going to put them into a little machine, and the machine is going to do the work of PCR. And the work of PCR is just heating and cooling. And so you say, all right, how does this work? Well, first step, you are going to denature your template, which means take double-stranded DNA and make it into single-stranded DNA. And yeah, in the first step, we're going to do this usually a little bit longer just to make absolutely certain that all of this gets done. I think more modern PCR protocols would try to shave a little time off of this. So you can usually go for a 30-second rather than 3-minute initial denaturation. Then you're going to run cycles. So step 2, 3, and 4, you're going to repeat over and over and over again, just like this says. It's written like a little computer program. And yeah, if you operate a thermal cycler, which is what that machine is down there, this is exactly how you set it up and how you program it. So you set your initial denaturation, which is going to be at 95 degrees, which is going to separate your DNA strands. Then you're going to cool down to somewhere around... Now this, you're going to vary this. So the lowest temperature you'd probably want to use would be around 50 degrees Celsius. The highest you would probably want to use is about 65 degrees Celsius. But anything less than 65 degrees Celsius is going to be a temperature where your Taq polymerase does not work. And so you can just wait and, carefully bind all the DNA down to the right templates. And so at cooler temperatures, you'll get DNA that potentially binds to the wrong place or a partial match. And we want to adjust the annealing temperature so that we can only really bind with perfect matching, perfect complementarity between the primer and the template. And so as you're designing your primers, you're going to figure out, you're going to calculate what your annealing temperature is for the primers. And you're going to try to make sure that your forward and reverse primer will have a very similar annealing temperature, because the whole reaction with forward and reverse primer is going to be in the same tube. And so, yeah, for this to work, they have to be nicely matched. And then once you have bound your primer, and hopefully not bound any primers to just junk sequences, hopefully you've got your annealing temperature about right. Then you're going to raise the temperature up again to about 72 degrees, and you're going to do your extension. So again, sort of time-saving modern protocols on really fancy machines. You can shave these steps down to about 15 seconds each for DNA denaturing and primer annealing. 1 minute's an older setting from back when the machines were a little bit slower at heating up or cooling down the PCR reactions inside. And PCR reaction, by the way, is redundant because PCR stands for polymerase chain reaction. So you just say PCRs, or the PCR tubes, or the reactions. Yeah. All right. And then after you have done step 4, extending the primer, the Taq polymerase is going to go at a rate of about 1,000 nucleotides per minute. And so if you're trying to amplify something that is 1,000 nucleotides, a minute is great. You usually want to leave it a little bit longer in case there's some of these Taq polymerases that run into trouble, lose their magnesium, go a little bit slower, whatever. So you pad it out, give it like 50% extra time, something like that. And then, yeah, you're going to go back and repeat step 2. So you're going to denature, anneal, extend. Denature, anneal, extend. You'll usually do this up to 40 times in one PCR. About the minimum you'd want to do it is something like 25 times, but 40 times is, yeah, usually if you're trying to amplify something that is relatively uncommon, or if you're trying to make a lot of product. And then after that, you end. You're done. Yeah. All right. Once you have finished PCR, you're going to take the tubes out of the machine, just like it says, and you're going to make a little gel. So you will have made gels like this in our Biology 1 lab. And so you'll be doing electrophoresis with, something in that lab. And so what you do is you'd make little holes in it. You'd put the DNA into these little wells, which are little holes that you make by putting a little plastic comb in there as you're allowing the agarose to go solid. And then you're going to apply an electrical field. The electrical field will push the DNA. And shorter pieces of DNA are going to move faster, longer pieces are going to move slower because they're being pushed through a spongy material. That's what the gel is actually going to look like. And yeah, they're like little pieces of spaghetti, basically. The real short ones can move rather easily through the spongy gel, and the long ones just keep getting twisted up and tangled and caught as they run. And then you're going to run out on the side something like a marker, which is going to have DNA of known sizes. So you're going to buy some of this from a company. And yeah, if you got the marker there, you can then compare the size of your unknown to the size of the marker. And you can figure out how big it is. And you can see whether or not you actually amplified the thing that you were hoping to amplify. So some lanes may not have a product if you did not get an amplification there. And so this is a good yes-no test. And we can also turn this into a quantitative test. That's what the next thing. So here's how you're going to do, you would do electrophoresis. You've got a little gel. You're going to put it into this tank. And the tank is going to have a little platinum wire at either end. And we use platinum because it's very good at conducting electricity. And then you're going to have a little box here, which is going to generate a current across this. And the current's going to be transmitted through the water. And there's going to be buffer there as well. And it's going to push the DNA along. We're going to push the little ions along. And at the platinum wire, you're actually going to do electrophoresis, and you're going to generate a little bit of oxygen and hydrogen. You're going to break down a little bit of that H2O, and that's going to be fine as well. You're going to see little bubbles coming off. So you're going to release nice oxygen into the world, but not enough that the hydrogen is going to explode or anything. It's a fairly tame, small process. And so, yeah. The gel is going to run from the black toward the red. I think of a DNA gel as like a budget. They tend to run in the red or toward the red. Yeah. So yeah, there you go. All right. And then yeah, the DNA is going to be clear. You're not going to be able to see it, but after you would run a DNA gel, you will be able to put dyes on that, and there are some dyes that you can just see under regular light, and there are other dyes that are going to be really sensitive that you'd be able to see under ultraviolet light. They would show up as fluorescent dyes, and they look really beautiful. Those are the ones that people tend to use because they are much more sensitive. You can see smaller amounts of DNA, and you can see it very clearly. Okay, and so yeah, you can compare your band size against some kind of what we call a DNA ladder over here, which has all the different sizes on it, and that's how it is. So there is a nice video that is going to be linked on Blackboard. You should look at that. That's going to walk you through the PCR process one more time. And the next step we're going to go to is going to be quantitative PCR. So we're going to figure out how to do PCR in such a way that you can tell how much product you made. And the answer is actually really easy. You just use a dye that sticks to the DNA, and the more DNA you have, the more dye you're going to get bound. Very, very simple. All right, that's the end of this first section.